So I came up with the title Nanoparticle-based mucosal delivery.
Can you speak through the notice?
Oh, this one. This one? Hello? Okay.
Like a stinger, you can actually take it.
Okay, so I'm focusing on a different route of administration. I don't think this has been covered at all during this course yet, but that is to use nanotechnology to facilitate delivery of RNAi therapeutics across mucosal surfaces. But I also want to try and give you an impression or a description, should I say, of my experiences setting up a spin-out company in Denmark based on the technology which I'm going to focus on, which is a very simple technology, which is based on the Polycatine Chitosan for delivery across mucosal surface. But I think, you know, the thing I want to stress is that, and I've got another title, Simplicity, the key to clinical translation. And we've heard a lot about elaborate designs of nanoparticle systems, but the way I see it is the simpler the better in terms of getting a product on the market. So you'll see that this system is pretty simple, but I think you'll also see that we've had some quite good success by using it. So as this is a nanomedicine school, I should set some educational goals, and that is that what I'll do is I'll describe systemic barriers and what is the motivation behind alternative routes of administration. So why do we need to deliver materials, for example, across the mucosal surfaces rather than injectables? I'll focus on mucosal barriers and delivery solutions. That will be half of the talk, and the other half of the talk will be utilizing the same technology for the treatment of macrophage-based diseases. So just, you know, to show you where we are, who we are and what we do. So Aarhus is in the peninsula that reaches out from Germany. It's on the east coast of Jutland, not far from Britain, I suppose, across the North Sea. We're based at the University of Aarhus, and the Nanoscience Centre, which was spawned out of the physics department at the university in 2002. Since those days, we've now got a purpose-built iNano house, as you can see here, that was opened in 2012, is it now? 2012. And as you can see, we've got 55 full professors, associate professors, assistant professors, 66 postdocs, and approximately 140 PhD students. But just to say that this does look a quite small nano house, but underground, in the basement, 2/3 of the area is actually underground. So a lot of the machines, etc., are buried beneath this building. So, you know, in years to come, will we be talking about nano, or will it be femto, etc.? I'm not so sure. I think that nano may come and go, but I think what we do need is interdisciplinary. And the former director of iNano in 2002, I think, was very forward-thinking, because the name iNano is the Interdisciplinary Nanoscience Centre. And I think that interdisciplinary is the key word. So in iNano, we can draw upon the combined expertise of physics, chemistry, biology, for example, all working together on projects. And interdisciplinary, to me, is not a scientist who branches out in a completely different field that's beyond his expertise. Interdisciplinary for me is to have possibly a house, a nano house, where you've got experts who are specialized in their particular subjects, who can contribute together in combined projects. I don't think scientists who branch out of their expertise or too far out works. I think interdisciplinary is sticking to your expertise and contributing that expertise to a combined project. Luna is our nanomedicine program. It's sponsored by Lundbeck, the Danish pharmaceutical company. and it's focused towards treatment of musculoskeletal and neurodegenerative diseases. And as part of that, we work on drug design, target validation, drug delivery, of which I'm prominent, and bioimaging, cell-based therapies, and preclinical models. But as you can see, we combine all these expertise together in these combined efforts. And just to say that this was established approximately 2 or 3 years ago, and it was, we got funding for about $50 million for this. Okay, so let's go on to a bit of the background and the motivation why we need delivery solutions for siRNA. Now, we do know that siRNA, let's just talk about conventional siRNAs for the sake of it, have a poor pharmacokinetic profile due to susceptibility to serum nucleases. Their size allows them to be readily excreted within a couple of minutes. You get non-specific accumulation, and they're polyionic, and so it's very difficult for them to cross the biological membrane, that is, the cell surface. In the early days, there was a lot of optimism for the use of siRNA in the clinic. Some early studies, the one by McCaffrey in 2002 and Sushek in 2004, in the McCaffrey one they used naked siRNA and showed that by hydrodynamic injection, so that's injecting quite a lot of volume of the material in the tail vein of a mouse, they could downregulate luciferase, that is, in the liver. In the Sushek work, they conjugated a cholesterol to the siRNA, and cholesterol is metabolized in the liver, and again they could see some therapeutic effects by downregulating ApoB, which showed then had knock-on effect of showing downregulation of systemic cholesterol. But we'll come back to this, but even in those early days, the liver seemed to be the primary target, or the target that was most easily approached, should I say. So naked siRNA didn't seem to work unless you had these artificial situations. So we had an whole range of nanocarrier systems, lipid-based or polycation-based. We work on a lot of different systems and elaborate designs as well. So we're part of this trend to invent the spaceship, should we say. But for this talk, I'll talk about a very simple, conceptually simple system that's based on a chitosan, a polysaccharide, that self-assembles with siRNA or nucleic acid, should I say, to form nanoparticles or polyplexes. These can also be called polyelectrolyte complexes and Polycatine-based systems, but we'll just call them polyplexes. But we knew there was problems with delivering conventional nucleic acids, or should we say naked nucleic acids. We thought that having these nanocarrier systems would overcome the problems associated with naked delivery. But I think in this one slide, you can see the complexity of delivering something systemically. So we can have a beautifully crafted nanoparticle. The previous speaker showed fantastic data of a well-crafted monodispersed system, and that's fine on the bench, but what really matters is what happens inside the body, inside the bloodstream. So if you inject a nanoparticle, and this really applies to most nanoparticles that are not surface modified, within a few seconds you can get protein adsorption to the surface, which causes aggregation. And this is one of the reasons why, systemic delivery of nanoparticles that are not surface engineered can find accumulation in the lung, because you get aggregation, so you have this nice nanoscale system that suddenly becomes a macro-scale system. And then it gets trapped in the first capillary bed, which is in the lung. So in the early days of polyethylene imine-based systems, they got aggregation and could actually get silencing in the lung. But obviously this is not transferable to the human situation. We can't have aggregation in the lung. But one of the main barriers to a systemic delivery system is the mononuclear phagocyte system, which is a series of circulatory monocytes and fixed tissue macrophages. I know one of the speakers was talking about Kupffer cells in the liver that are basically designed to pick up foreign materials, entities floating around in the body, of which nanoparticles are seen. They're phagocytosed. They're elaborately designed to destroy this engulfed material. And this is one of the reasons why we've had so much success with nanocarriers when we deliver to the, or should I say, siRNA delivery to the liver. Because one of the main sites of these phagocytes, or the phagocyte population, is in the liver. Plus, it's highly perfused. It's got a good blood supply. But, you know, it's no surprise that people are getting delivery to the liver. The question is, are they getting delivery to the right cell population? And I've not really seen definitive evidence from anybody working in the field on the ratio of accumulation in, say, the Kupffer cells, which are the phagocytes, and, for example, the hepatocytes, which are the target cells. If we can reduce this interaction with the MPS system, we still have to get extravasation across the endothelial barrier and then into the cell. So you can see there's a myriad of barriers when you try and inject something systemically. We can surface engineer the surface of particles, so pegulation is the common practice for this. You can have different graft densities, so you can have different configurations on the surface. The most notable configuration is the one in B, which shows a brush border, a brush configuration, which does show reduced interaction with phagocytic cells. But then you have the stealth dilemma. It's one thing reducing interaction with a cell type such as a phagocyte, but then you've got to retarget that particle into a target cell. And that is the problem. Where do you tether the ligand? Do you put it on the end of the peg? If it's a peptide, it can then be immunogenic. Or do you bury it deep down in the brush border, in which case it's not accessible to engage with its target? So from my point of view, using a nanocarrier system that's pegulated is not the ideal scenario that you want to go for. This is not the full shebang of clinical trials, but I've focused on one of the main players in the RNAi field, Alnylam, and this is sort of a recent search I did. And I think what's the most striking thing is that in 2014, where we are now, that most of the indications which they're working on are focused on the liver. And in fact, on their website, one of the criteria is that they want a liver target. And that's primarily because accumulation in the liver. They've had problems directing the nanocarrier to any other sites, as we all have. And so now they're basically trying to make the most of where they are, and that's whatever nanocarrier system they're using. They've used lipid-based, now they're using sugar conjugates. It's still accumulating in the liver, which really reduces the old span of, or should I say, the range of diseases which you can treat with any RNAi-based therapeutic. Interestingly, there was this naked siRNA without any nanocarrier formulation, which was against the respiratory syncytial virus, that actually reached phase 2B. And this was injected directly into the pulmonary tract for delivery to the lung. And they seem to get obviously quite nice data early on, but this, I think, has recently been withdrawn. So now they're solely focused on liver indications, as far as I know. Techmura, which is also working on lipid-based systems, again are working on liver targets. I've just put this Calando, which was one of the first polycationic polymer systems to reach clinical trials. It came out of the lab of Mark E. Davis in Caltech, where he was using an elaborate system with a cyclodextrin, where he sunk adamantine with pegylated and transferrin for targeted delivery to tumor cells. But as far as I know, this has also been withdrawn. So where we are now is that the indications that we're focusing on is in clinical trials is mostly the liver. So I've put a focus on liver indications predominantly due to physiological consequences after IV injection. That can be phagocytic uptake, this can be accumulation due to highly perfused blood supply, it can be the disrupted endothelial you find in the liver, it can be picking up by hijacking the apoprotein E, I think, that then goes as a matter of consequence to the liver. So I would propose that alternative strategies are required and simple strategies. So here I go with this very simple system, which is a Polycatine-based nanoparticles for siRNA. We use a polycation that electrostatically interacts with the siRNA to form a nanoparticle. Entropy-driven, it's an highly efficient process, but this simple system actually installs many properties into the nanocarrier. It's a self-assembly system, so to some degree the siRNA is protected and entrapped within the system. It's nanoscale, so it's small enough to extravasate across certain diseased tissues. It normally has a net positive charge, and when you make these systems, they're more stable when you have an excess of the polycation, which has got the predominantly amino groups, so it's a positive charged entity which interacts very well with the cell surface. You can control the functionality, which is the pharmacokinetics, cellular targeting, uptake, route and RNA release, by the design of the polycations. So you can do this by synthetic systems. But I would say that this has got clinical translation potential because it's a very simple system. We're not relying on any post-modification. I won't go through this slide, but just to say there's an whole array of polymers, both synthetic and natural. I've focused on ones that have been used in vivo, that have been used for delivery of siRNA. When I say siRNA, this could be the conventional siRNA, or this could be precursor microRNAs, primary microRNAs, for example, but we'll just stick to the siRNA as a general term. We can change the polymer architecture, especially for the synthetic systems. And you can see that you can change the functionality by using different polymer systems. I won't go into detail on this, but just to say that it's versatile and flexible, even though it's conceptually very simple. So here we go. This is a simple system that I propose. This is the Chitosan system, which is, chitosan is a polycation derived from chitin, which is found in the exoskeletons of insects and the shells of crustacea. By deacetylation, you install an amino group and a glucosamine. And it's this amino group that facilitates this interaction with, for example, a polyionic nucleic acid. Very simple. It's the second most abundant polymer, natural polymer, should I say. But what people have found, even before the days of siRNAs, it's been used for its mucoadhesive properties, and that is that it facilitates interactions with glycoproteins that are found in the mucus that lines the mucosal surface on the epithelial cells. I'll go into a bit of detail on that. It's also been shown to transiently open, so for a short period of time, open the tight junctions between the epithelial cells at the mucosal surface. The system itself is this polycation plus siRNA into a nanoparticle. And just to say, although this is a conceptually very simple system, it took about 2 years to optimize the type of chitosan to be used for a system that would work biologically. So what you can change is the molecular weight and the degree of deacetylation, the amount of amino groups, and the polymer backbone. So the two main attributes of a chitosan system is that it's mucoadhesive. So if we just look at the mucosal surface, so the mucosal surface is the lining of the respiratory, gastrointestinal, genitourinary tract. And if you look at the epithelial surface, you've got tightly packed epithelial cells that vary in morphology dependent on the anatomical site, but normally they're tightly packed together. In the lung, they display these ciliary outpushings which push material away. But then overlying these epithelial surfaces, you have this gel, this physical mucus gel, which is primarily composed of mucin glycoproteins. But just to say that the mucus layer that overlays these surface is in two layers. So you have a more viscous fixed layer at the bottom, underneath, that borders the epithelial surface. Then you have a more watery fluid system at the uppermost surface. And it's in this uppermost surface that's brushed away and replenished, depending on the site in the mucosal surface, it's replenished every minute, few minutes, or every hours, or hours, should I say. If we zoom in on the mucus itself, the main constituent of mucus is these mucin glycoproteins, which are polymers that form a network. And this network forms pores in which material has to penetrate through the pores in able to reach the underlying epithelial cells. So when we think of a delivery system, we need to consider this microstructure that's composed of these glycoproteins. The other main attribute of Chitosan is that it's been shown to enter macrophages more avidly than a lot of materials. Now we know that particles have a predisposition for phagocytic capture. This is one of the problems of a delivery system. It's recognized and captured by phagocytes. But mannose receptor on the surface of the macrophages engage with the polysaccharide and the chitosan, and you get more uptake into these cells. And I'll go into that. Just to say that it's one thing getting uptake of a nanocarrier system into a macrophage, it's another matter to actually get, for example, gene silencing, because there's an elaborate network of materials, of lysosomes that can destroy and break down ingested material. So we started this work, let's say in 2004. For the next 2 years, we gathered data, we optimized the system, and then I'll show you some of the data that we published. But what happened was, the series was that we came up with this idea of using siRNA with Chitosan. Now, Chitosan had been used extensively for the delivery of peptides and also plasmid DNA. But we were the first to actually use it for siRNA, and we secured a patent on the siRNA. So you can see that although this system had been used extensively for other molecules, we were quick on securing IP on this technology. And this is important moving forward if you want to establish a spin-out. So we didn't publish at that point. We prepared a patent application. So we formed a company called Nanoference, based on the Chitosan system, based at iNano, the Interdisciplinary Nanoscience Center. And as I said, the actual spin-out was derived from the technology surrounding the Chitosan system at the university. And just to say, I don't know what it's like in other countries, but in Denmark, if you do some scientific work in the laboratory, the patent is owned by the university. So what we have to do is, as soon as we thought that we had a novel idea, we had to approach our technology transfer office and prepare a patent disclosure. And this is a requirement as a university worker in Denmark, that you have to consult with a TTO and offer them the chance of taking on the patent. If they don't want the patent, then the inventors themselves can take it forward. But they did like the patent, they did their due diligence and thought that there was some way forward with it. So actually, the patent is actually controlled by the university, but it's partly owned, a third for the university, a third for the inventors, and a third for the department. So if we make any money out of this, then iNano itself will get something back. We launched it in 2010, and this was based on securing seed capital from two Danish companies, one Nova Seeds, which is a derivative of Novo Nordisk, the big pharmaceutical company, and another small investment company, Seed Capital. And they invested near on $800,000 into this spin-out, so this was Seed Capital. But to convince them about this technology, so we filed a patent, and then the TTO actually reached out to potential investors. We had clinics where we met investors. So the university itself holds sessions where investors can meet potential inventors for potential investment. And they were happy that we had IP. They don't want to invest in anything that's not protected. And they were also happy that we'd published in good journals. For example, a lot of this work went in Molecular Therapy, which is quite a good journal in the gene therapy and RNAi field. So those two, plus the fact that they like the enthusiasm of the inventors. It's amazing that, you know, they put a lot of, they place, well, let's just say that they feel that in order to invest, they must feel the enthusiasm from the inventors. And we were very enthusiastic about this. So I did a lot of this initial discussions with Nova Seeds and Seed Capital. They liked the way that we worked, and so they gave us this investment. And the company itself, the founders and myself, and I was placed in a CEO position. Jan Kems, a professor who's an expert in RNAi, and Fleming Basenbacher, who was the director of iNano, who was actually involved in some of the scientific work. And then what we did is we base our company actually within the labs of the university, but what Nanoference does is it sponsors university people. So the people working for Nanoference are actually employed by the university. We just sponsor that funding. And again, what they liked about us, the investment company, we had a strong patent portfolio. We had approximately 4 patents surrounding this technology based on different routes of administration and different treatments. And there we go. So just to say that you can attract interest by using a very simple system. I think one of the important things to consider when you utilize a nanocarrier system is you have to understand the biological barriers. And I showed you the 2 properties of Chitosan. And if you know those properties and you know also the limitations, you can target that particular delivery system for a certain application or route of administration. And as I said, that we feel that it's a mucoadhesive system that can penetrate these mucosal barriers. So it was obvious that we'd focus on pulmonary delivery to the lung. I'll just describe some of the work we did, extensive work with an EGFP model system, and work we're continuing in cystic fibrosis. So we worked with this transgenic mouse that expressed EGFP. Here we have the simple nanoparticle image with atomic force microscopy. And just to say that we administered these particles intranasally was over a period of 5 days consecutively into the nose of the mouse. And then after a period of 5 days, we sacrificed the mouse and harvested the tissue. And remember, we're focusing on delivery to the epithelial layer of the mucosal surface. And you can see here in the control tissue, we've got the EGFP expression in the epithelial cells that border the bronchi in this case. And with the EGFP-specific siRNA delivered in our Chitosan system administered intranasally, you can see that we downregulate the EGFP expression. And we've co-stained with DAPI, a nuclear stain, to show there's still an intact border. So we're showing that we have got knockdown. We then developed this system into an aerosol because we want to reach possibly down into the deep lung. One of the problems of these biomaterials that are mucoadhesive is if we apply them into the nasal cavity that they're going to stick in that region and they're not going to reach down below. So we nebulized these systems, we optimized that procedure, and we could, I don't think you can see it here, but we had an siRNA that was fluorescently labeled, and we can show extensive distribution in deeper regions of the lung if we form an aerosol with this system. We also did, in collaboration with the Max Planck Institute in Berlin and the lab of Thomas Meyer, who's working on RNAi interference approaches for treatment of influenza. Initially we did some in vitro studies to show that we could silence a conserved site in the influenza, which was the nuclear protein. We then used that siRNA in an animal model that was infected by influenza, and we got some degree of knockdown, or should I say reduced infectivity in the lung using this was an LNA stabilized siRNA system. This work is ongoing, and actually we're working now on using targets of the host cells. So you know that viruses can hijack host cell processes and pathways. And in conjunction with Thomas Meyer, we're now focusing on using siRNA to interrupt some host pathways that are utilized by the influenza virus. This is some recent work, and we've only just really moved into this, but this is utilizing this very simple technology for the treatment of cystic fibrosis. So cystic fibrosis is caused by a mutation in the cystic fibrosis transmembrane conductance regulator, which regulates the transport of anions, sodium, should I say, sodium and chloride across the epithelial surface, which is needed to maintain the homeostasis of the mucosal fluid. If there's a mutation in this gene, you find that you get a retention of the mucus in the respiratory tract, and you get a buildup of mucus. It's not watery enough, in effect, to be moved from the site, and you get a gradual buildup. And this is a terrible disease, because even now, between the age of 20 and 40, the victims, should I say, die, or they need a lung transplant. And there's no real cure for cystic fibrosis at the moment. So our therapeutic strategy was to target this AHA1 gene. Now, it's been shown that this mutated CFTR gene, the regulator, although it's misfolded and mutated, if it reaches the epithelial surface, it can still have some functionality and reduce the cystic fibrosis condition. And AHA1 is actually a mechanism by the cells to remove this mutation. So it's a degradation pathway that actually is there to repair the mutation. But if we can knock down this degradation pathway, we can still utilize this mutated gene, which would then still have some functionality and reduce the buildup of the mucus. I must say that, it's one thing going straight in vivo, but obviously when you do RNAi work, you've got to screen, for example, different sequences in vitro. So there's no way that you can go straight in vivo and do these systems. So what we've got here is we use different designs of siRNAs, designs that would reduce off-target effects or immune stimulation, also stabilized siRNAs targeting the AHA1. And we used a human CUF1 cells, which are pulmonary epithelial cells that express the AHA1. And then we looked for knockdown. And you can see that using our very simple system in vitro, we can downregulate this target gene. Now, moving forward, there's no real good murine model for cystic fibrosis that mimics the human condition. But what they have found is the pig model does. And in collaboration with Paul McRae at the University of Iowa in the USA, they've got a pig model that has the common mutation, which is a delta 508, that basically mimics the human condition. So our way forward is to try and get delivery in pigs, because at the end of the line we've got a very relevant model to test our system that mimics the human condition. But we don't want to just look at cells on a plate. We want to try and replicate the liquid air interface that you find in the pulmonary tract as a model for delivery. So what we've done is we take the trachea or bronchi from pigs, We isolate the cells and we digest with collagenase. We isolate the epithelial cells, and then we basically form a liquid-air interface model that mimics the pulmonary tract. If we do TEM, you can see that we've got these types of cells, but what they also show is that we've got goblet cells, and goblet cells are the cells in the mucosal cells that produce mucin. So if we want to replicate the barrier that we'll combat when we go in pigs or humans, we've got to have a cell line that expresses mucin. And we can see that we get goblet cells, and we do actually see mucin produced in these cells. This is actually an elaborate in vitro model, and in itself it's pretty difficult to silence. We have done that. We've used a housekeeping gene, HPRT, that's found in pig epithelial cells. And using different N/P ratios, the ratio of the nitrogen to the phosphate in the formulation, you can see that we get some degree of knockdown. But this is a tough model to show knockdown in, but it's looking promising that we can isolate this model and then we can show knockdown. The next step is to show delivery of our system in a pig model. And we've got good collaboration with medics at the University of Aarhus in the cardio unit that use pigs quite extensively. And in here we're actually applying our delivery system directly into the trachea of the pigs using nebulizers. First steps is to use fluorescently labeled model systems such as latex particles, just to see what size will get good deposition into the lung. And you can see that we can show with this system good deposition. Just to say that this was imaged with one of these IVIS bioimaging devices. So the future work now is we can show that we can get knockdown of the HAHA1 gene. We can show that we can silence and get transfer across a liquid-air interface model. The next thing is to show knockdown of epithelial markers in primary pig airway epithelial, the liquid-air interface. Then to show knockdown in R1 in the pigs. Knockdown of epithelial markers in the pig, and then eventually knockdown of the R1 in CF pigs to see if we can reverse the CF condition. And this is where we are at the moment. I'll then now switch to oral delivery using this conceptually simple system. Now you would imagine that an electrostatic interacted system that's delivered orally would be susceptible to breakdown. We thought that, and so we've tried some initial studies where we're focusing our target disease for the delivery by the oral route is inflammatory bowel disease, which is an inflammatory response that's primarily caused by the production of the pro-inflammatory cytokine TNF-alpha. There's two types of, well, there's an whole range of inflammatory bowel disease, but there's the Crohn's disease, which is located in the small intestine, and then you've got the ulcerative colitis, which is more in the colon. And just to say, and I'll show you why, but we're trying two approaches: conventional siRNA approach as a therapeutic, but also locked nucleic acid antisense, and I'll come on to that. We did some initial studies where we, by oral gavage, administered the Chitosan system with a siRNA, a model siRNA. And then we harvested the tissue at 1 hour or 5 hours in different locations in the alimentary canal. And as you can see, when you compare it with the siRNA, naked siRNA administered material, that by Northern analysis, we can see deposition throughout the whole of the intestine. It's quite surprising that we've got stability in the stomach. But you can see that this very simple system can be administered, albeit by oral gavage. So it's an artificial approach in a way, because you're flushing down with quite a large volume into the stomach that's flushing it through. But we are getting preserved integrity of our siRNA when it's within this very simple system. We then extended these studies to look if we could get translocation across the intestine to the peripheral tissues such as the kidney, spleen and liver. We looked with Northern analysis and also qPCR, and what was surprising is dependent on the N/P ratio, so when we had a high Chitosan content in the NP120, you could see that we could observe or detect the siRNA in the kidney. And this is amazing, really, that you can have this very simple, unstable, you would think, system that can be administered orally, but it can reach these peripheral tissues intact. We then tried to investigate the uptake at the epithelial surface, so we used fluorescent siRNA. We used in situ gut loops where we ligate either side of a certain region of the intestine and inject fluorescent material. And what we did see is this fluorescent halo around the epithelial surface. Now the problem with these polyelectrolyte complexes is when they're in a particulate form, you can observe them under a fluorescent microscope. But if they disassemble and the siRNA is naked, should I say, and liberated, it's very difficult to pick up the punctate fluorescence. So we weren't sure actually what was going across. We know that the material was reaching the kidney, for example, so the siRNA does reach that site. But is it going as an intact nanoparticle or is it going as a free siRNA? You would think not, because the siRNA shouldn't cross these borders. Just to say that we're working in collaboration with the lab of George Collios, who's a pioneer in TNF-alpha therapeutics, and he's got this transgenic mouse that overexpresses TNF-alpha that exhibits symptoms of Crohn's disease. And the idea next is that we're going to use a TNF-alpha siRNA in our Chitosan system and see if we can reverse the inflammatory bowel disease condition. That's ongoing. But we're intrigued on what's actually happening at this mucus nanocarrier interface. Is the nanocarrier going over intact, or is the siRNA moving unassisted? We did some simple studies where we had cell lines and we put the particles on top. These are these polyelectrolyte complexes fluorescently labeled, and they could reach the cell surface as you would expect. When we had the cells such as goblet type cells that secrete mucins, you could see that, well, I don't think you can see it clearly, but there's a disruption of the particle and there's a definite barrier as you would expect. So we feel that, you know, there is definitely a barrier to penetration by these nanoparticle systems. We used a very simple system where we used a fluorescent activated reporter assay, whereby tethering or conjugating fluorescent tags to the terminal of the 5' end of the siRNA, that we found that when they were in close proximity, that you had a self-quenching effect. So when we had the siRNA in a free form, that we had fluorescence. When we condensed it down into a polyelectrolyte complex, we had a self-quenching effect. We saw a decrease in fluorescence. And as you can see here, we had the siRNA that was labeled with a fluorescein type tag. And then as we increase the N/P ratio and we form a complex, you can see we get a reduction in fluorescence. And I'll go into detail on why we need this reporter system. So the idea is that we wanted to see if the mucins, which is part of the mucus barrier, can actually disassemble these polyplex systems. A way of doing that is to incubate them with different concentrations of mucins, and then to see if we can then increase the fluorescence, which shows that we've got disassembly of the particle. And lo and behold, as we increase, this was porcine gastric mucins, we could see that we get an increase in fluorescence, which indicates that the Chitosan system is disassembling. We saw this also on gels. We could see liberation of the Chitosan system when we had a high content of the mucins. We wanted to investigate more thoroughly this interaction of the mucin barrier. So in collaboration with Katerina Rebeck at MIT in Boston, she's got a microfluidic system where she has mucin hydrogel, and then she flushes materials across it, and then you can investigate the interaction at this mucus or mucin interface. We use porcine gastric mucins in this case. And then we're looking for diffusion of the particles. Remember that we'll detect the siRNA when it's liberated from the Chitosan system. So we'll be able to investigate not only the movement of the siRNA, we'll actually investigate the form. Is it a particulate or is it disassembling? And we investigated two different types of systems. So here we've got an NTP60, NTP5, and we're looking for the movement. You can't really see it clearly here. These are depicted by arrows. We can see the movement of free siRNA when the particles reach this interface. And surprisingly, we got greater movement of the particles that were composed of the higher chitosan content. And what we think is happening is the chitosan, or the excess chitosan in these systems, is interacting more so with these mucin glycoproteins, the sialic acid predominantly, interacting and ripping the polyplex apart, allowing movement of the particle in there. We also tried the diffusion of naked siRNA. So we wanted to see if naked siRNA, unassisted, could migrate across a mucin barrier. These are just fluorescent beads that are used to measure the diffusion of the system. But you can see that over time we get a diffusion into the mucin of our fluorescent siRNA. And this is interesting. That means that naked siRNA can actually cross a mucin barrier. The problem is that naked siRNA is not very good at crossing cellular membranes. It's polyionic. It's quite big. And so we have a problem. So the proposed model and a new design is that we have siRNA nanocarriers. They interact with the mucin glycoproteins, they disassemble, and then we get movement of the free siRNA. So what we're thinking now is we can utilize this process for mucus-mediated triggered disassembly for cargo release. So only when they come in contact with the mucus barrier do these nanoparticles disassemble and release their cargo. And the way forward is to use a different RNAi therapeutic that can enter cells unassisted. We know that double-stranded siRNAs, conventional 21-mers, do not cross the cellular membrane, but antisense does. So we've used locked nucleic antisense. So these are called Gapmas. So they're essentially antisense DNA molecules, single-stranded, that are flanked by locked nucleic acids that stabilize the structure. And these single-stranded materials have been shown to enter cells unassisted without any nanocarrier system. And no one really knows what the mechanism is. What is the driving force of these? They are still polyionic to actually cross. Maybe they're small size. But you can't really see it here, but we've tested the ability of these antisense molecules to cross macrophage cell lines, CACO2, which is epithelial models, but also we've taken primary murine epithelial cells, intestinal cells from mice, and we can see very good uptake. It's not very clear on this picture, but believe me, these things go into these cells quite avidly. So our new system that we're going to develop is a Chitosan-based system incorporating these types of materials that can, once released, enter these epithelial cells. Just to say, a number of techniques we're utilizing at the Nanoscience Center to investigate the interactions with mucus and mucins. So we're using atomic force microscopy, obviously, to see the morphological changes. We're doing a lot of work by applying different types of biomaterials other than chitosan to these mucus, the mucins that interact with the mucins and can disrupt this micromesh and can maybe open up the pores, which allows greater penetration by nanocarriers. So we're doing a lot of work. We apply different types of biomaterials. We're looking at the morphology of this mucin network. We're also looking at the bioadhesion of biomaterials as alternatives to chitosan by using force pulling techniques. We've also developed a very simple system utilizing a very common piece of equipment, the nanosite, which is used as it's a particle tracking analysis technique that's used to measure particle size. It works by shining laser at particles that move by Brownian motion and deflect the laser, which deflect the light, which is then captured by a camera, and it's routinely used to measure size of nanoparticles, an alternative to photon correlation spectroscopy. But we've developed this system to look at the movement of particles in mucin hydrogels by looking at the diffusion coefficient. And what we can do then is to apply different biomaterials to these mucin hydrogels and then to see if we can alter the movement of the nanoparticles as a prelude to using these types of systems therapeutically. And this is just showing that by using different systems we can alter the porous network, which enables greater uptake. We're also using particle tracking using confocal microscopy. We've got a well-in-a-well technique where we use a mucin hydrogel. We apply any types of particles onto the surface fluorescent, and then we track by confocal microscopy through the mucin hydrogel the movement of the particles in real time. We're doing a lot of work in situ gut loops, so this is where, as I said, we do a laparotomy, we open up the animal, we isolate a section of the intestine, for example, that may be of interest for uptake. We inject materials that then are allowed to penetrate the mucosa. We isolate or dissect that system, and then we can isolate the different cell types, and by confocal microscopy or flow cytometry, we can visualize if these particles enter. How am I doing for time? Is it keep on. Okay, so I can, here's something completely different, but it's utilizing the second property of Chitosan. Remember, mucosal delivery, but also phagocytic capture. So we want to exploit this predisposition of Chitosan to be captured by phagocytes. So I've got it here again. The mononuclear phagocyte system is a series of circulatory and fixed tissue macrophages that recognize, capture, remove nanocarriers from the body. This is bad. The flip side is to exploit this for the treatment of macrophage-associated diseases. So we're using a natural stealth approach where we want to basically mimic what happened with the Trojans when the Greeks entered the city of Troy. That they harbored inside a device that was then, they were undetected, they could enter into the city walls. Using the same concept, we want the phagocytes to pick up our Chitosan systems and then let the macrophages themselves migrate throughout the body. We know that these polyelectrolyte complexes are almost useless if you inject them intravenously. I don't believe that you can use polyelectrolyte complexes, even the ones surface modified, for systemic drug delivery. I think you've got to use them for mucosal delivery. If you inject them intravenously, they're going to aggregate within minutes and you're going to get macro size systems. The idea is that the macrophage pick them up and they move undetected. I'm going to show you some preclinical. We've worked on two different preclinical models. One where we use Chitosan system for the treatment of radiation-induced fibrosis, and I'll get onto that, and one for rheumatoid arthritis. Both these diseases are predominantly caused by the production of the pro-inflammatory TNF-alpha, tumor necrosis factor, which sets off a cascade of inflammatory responses. We've shown that our Chitosan system containing an siRNA targeting the TNF-alpha can downregulate the TNF-alpha levels in macrophages. So, as we expected, when we incubate a fluorescent Chitosan system containing siRNA, that's the red system, with a macrophage, it gets captured. We've got a DAPI-stained blue nuclei. It's one thing getting it into the cell, as I said before. Can we actually knock down genes once it's inside there? And we can. So we used our Chitosan system with the TNF-alpha siRNA. We took peritoneal macrophages, so they were primary cells from the peritoneum of mice. We stimulated them with lipopolysaccharide incubation. So we get these cells kicking out large amounts of TNF-alpha, and we normalized everything else to those. And you can see that when we have a nanoparticle containing TNF-alpha, after 24 hours we get knockdown, compared to the Chitosan system with a mismatch control at 24 hours and 48 hours. So this, we didn't really think that we could utilize our Chitosan system for knockdown of genes in macrophages, but this was remarkable to us that we could deliver something into a macrophage that's there to destroy materials, and we could still get the siRNA to work. And that then stimulated us to use them for macrophage-associated diseases. So I've got it here. Chitosan siRNA system can enter and silence genes in macrophages. This opens up anti-inflammatory therapeutics using this simple system. Rheumatoid arthritis, chronic inflammatory condition, 1% of the population, mainly women in the 40s. It's manifested as a destructive joint disease, although it's a systemic disease in essence. And it's, the main culprit is the inflammatory cytokine TNF-alpha. We know that macrophages produce the TNF-alpha, and they play a prominent role. But importantly, we know that the peritoneal macrophages are recruited during states of inflammation, even in the joints. So it's a reservoir of macrophages in the peritoneal cavity that are recruited at heightened immune states. And we didn't really target rheumatoid arthritis, but the more reading we did around it, and we found that peritoneal macrophages were implicated in the disease, and we could knock down TNF-alpha in the macrophages, then we had a therapeutic strategy that peritoneal macrophages were the cellular target for anti-TNF-alpha siRNA intervention therapy in rheumatoid arthritis. And the strategy is this. We know that these polycatinic systems are positively charged. If we inject them into the bloodstream, they're going to aggregate by serum-induced aggregation. In the peritoneum, there's no serum, but there's lots of macrophages. We know that they get picked up by the macrophages. The idea then is that we inject them into the peritoneal cavity. It's a serum-free environment. We don't get aggregation of our system. We exploit this capability of macrophages to engulf the nanoparticles, and we also exploit this added effect of the mannose receptor on macrophages that engages with the polysaccharide. And the idea is that they go inside these macrophages, the macrophages are then recruited during the inflammatory state, and it carries the nanoparticles to those particular sites, the Trojan horse effect, we could say, natural stealth. We did some studies where we injected we injected fluorescent siRNA Chitosan particles into the peritoneum of mice. After 2 hours, we harvested those peritoneal macrophages, and we could see that these fluorescent nanoparticles were collecting in these peritoneal macrophages as expected. Working with Mark Belke from Integrated DNA Technologies in Iowa, who are working on DICER substrate siRNAs, 27-mers. We use a 27-mer against TNF-alpha, either in an unmodified form or with a 2' oxymethyl modification, which blocks the toll-like receptor engagement of double-stranded siRNA and reduces the immune stimulation. But just to say, what we did is we injected our Chitosan anti-TNF-alpha siRNA into the peritoneum. We harvested the peritoneal macrophages after 2 hours, and then we measured the TNF-alpha levels, and we got some degree of knockdown compared to the mismatch control with both the non-modified and the modified. But you must consider that this experiment is very difficult to show good knockdown because we're injecting, and then after 2 hours we're harvesting those macrophages. Now, it's known that when macrophages engage with a nanoparticle, they're activated and they move away from that site. It's a known phenomenon that macrophages that pick up nanoparticles in the peritoneum then migrate from that region. So we're injecting these particles, and possibly the macrophages, a good population of those macrophages, are moving away, so it's very difficult to then harvest them and show the knockdown. One of the considerations was to use this 2'-oxymethyl modification. The reason behind this is that we didn't want to initiate innate immune response to this 27mer, such as type 1 interferon, and as you can see with the modified form we got a reduction in this response. So it seems, we could use a modification that doesn't show this non-specific effect, off-target effect, should I say. Moving on to the preclinical work. So we worked on a model, type 2 collagen-induced arthritis in DBA mice. And what we did is you induce this, this condition by subcutaneous injection of collagen type 2, over a period of 28 days. So what we do after 28 days is we measure the swelling, which is a measure of the inflammation in those joints. At day 0 here is actually day 28. So at day 28, we can correlate the size of the swelling to an arthritic score. And you can see that after intraperitoneal injection, so we're not applying this material directly into the joint, we're trying our therapeutic strategy of picking it up by the macrophages and moving to the joints. You can see that we get some degree of arrested development of the swelling with the Chitosan system with the TNF-alpha. This is just a dexamethasone steroid control and anti-inflammatory to show that the animals were responsive to treatment. But we did get some degree of arrested development. But it was more apparent when we took the paws and did histological analysis. So one of the, if we just move to this image here, this is sort of the natural state of an arthritic joint. So you get disruption of the cartilage barrier, you get a high degree of infiltration of immune cells, such as macrophages, lymphocytes, and you can see you get a total destruction of this area. And this is what was seen when we injected our nanoparticles with control siRNA. When we had the dexamethasone positive control, we saw an intact cartilage layer and no immune activity. When we injected the nanoparticles containing the siRNA against TNF-alpha, we got something that was likened to the dexamethasone. And this was remarkable because we're injecting into the peritoneal cavity and we're showing effects in the pores. I think I can just go over this in a couple of minutes. The second model that we've tried this strategy with is radiation-induced fibrosis. So one of the drawbacks of radiotherapy for cancer is that the radiotherapist has to limit the amount of radiation that they can administer, because a common condition is fibrosis that can manifest itself up to 2 years after the radiotherapy. So to ensure that in 2 years' time the patient doesn't get this condition, they reduce or limit the radiotherapy. So it's sort of a drawback in radiotherapy treatments. Fibrosis is predominantly caused by pro-inflammatory responses orchestrated by TNF-alpha. It causes tissue remodeling, hardening and deposition of collagen, should I say, which causes these types of conditions where you get these strictures where it's very difficult to move because you've got the fibrotic tissue. And there's actually no treatment for this, and it's quite a common late side effect of radiotherapy. What we did is we had a mouse model where we could irradiate the hind leg and induce this radiation-induced fibrosis over a period of time. In humans it can take up to 2 years. In mice it can take up to 9 months. So it was a long experiment, but again we used a Chitosan system containing siRNA against TNF-alpha, exactly the same as the rheumatoid arthritis, intraperitoneally. And we could see that when we inject, so the actual model itself, the way that they give it a fibrotic score is when you irradiate and they get this fibrotic condition, you get reduced movability or flexibility of the of the hind leg. And this is due to this scar tissue, this fibrosis. And you can correlate this to a score, and you can see we get this as a severity of fibrosis. And you can see that when we had the control nanoparticles or mismatch control, that over a period of months, that these animals did in fact show signs of fibrosis. Remarkably, when we gave these systems with the TNF-alpha intraperitoneally, it was a flatliner. And these were quite high groups, 11 animals in this group. And we could see that we could stop this, this condition. Because we administered them up to 9 months, because we had to ensure that, you know, it was we could stop the late onset of fibrosis, we administered these particles up to 9 months, which was 78 intraperitoneal injections. So it gave us an opportunity to look at the toxicity of these systems, if you imagine the amount of injections we gave. And compared to the control mice, which was given no formulations, and the nanoparticles administered over a 9-month period, in all the major organs we saw no histological evidence of toxicity. A final slide. We think that our nanoparticles are being picked up in the peritoneum by the macrophages, and they're moving from that site to the inflammatory site in both these animal models. We wanted to prove that. So we did an experiment where we had fluorescently labeled siRNA in a Chitosan system, and then we injected intraperitoneally these systems into the mouse, and then we tried to visualize the trafficking of these particles after irradiation. So we're using the radiation-induced fibrosis model. In the non-irradiated mice, you could see that there was no movement of the particles from this region. However, in the animals that were irradiated in the foot pad, you could see over a period of hours that we got movement from this intraperitoneal cavity into the foot pad. And we do know that the inflammatory state, in mice, the acute phase is exhibited at day, 13. So it was remarkable that when we tried to traffic the movement of these particles maybe at day 8, 5, even after irradiation, we got no movement. But it was only when they exhibited the inflammatory state at day 13 that we saw this movement that links it with the inflammatory state. We then localized, we did histology on the tissue, and we used macrophage markers, and we could see there was some accumulation. It's very difficult to see on here, but histologically there was evidence that there was co-localization of the system. The difficulty is actually knowing if the particles moved within the macrophages during this transit period, or they moved unassisted and then were picked up by macrophages locally. That's something we haven't proved. Okay, just to say, this is an interdisciplinary piece of work, and you can see in the blue this is iNano. So a lot of the work's been based in molecular biology, and a lot of the work has been in collaboration with Jan Kems, who's an RNAi expert, and he's led or is leading the pulmonary delivery side. We've got numerous people working on that, but just to say that we've been very strong in the physicochemical characterization, atomic force microscopy with the physics part of iNano. But it's very important in this interdisciplinary project to work with medics, and all these preclinical models have been in collaboration with different medics on the project. I have to thank people at IDT and Iowa that have worked on the different constructs, the siRNA, the liquid air interface model, the pig model, Thomas Meyer, who's worked on the Influenza. All these are ongoing collaborations, Katerina Riebeck at MIT, and we're in collaboration with different companies, Novozymes in Denmark, and also Chitosan in Belgium, that are supplying different biomaterials. Thank you.